In this video, we are going to describe the rearrangement hotspots in the human genome. Now, we are going to speak about rearrangement hotspots in the human genome. So, the rearrangement hotspot theorem says to us that there are fragile regions in the human genome. So, if the random breakage model is correct, and rearrangement applied to singular chromosome will produce approximately 2n synteny blocks. Why? Because each arrangement will start in a different position, because the breakage is random. So, we have 280 human mouse synteny blocks. As a consequence, we should have 140 two breaks on the human mouse evolutionary path. But the two break distance theorem says to us that there are at least 245 two breaks on this path. So, we have a contradiction between the random breakage model and the two break distance. So, one of our hypotheses was wrong. Which one? We are going to reassess the random breakage model. The point is that if the RBM is wrong, we would have an exponential distribution, but in reality, we will have different distribution, different hypotheses compatible with these distributions. So, a non-random breakage model means a breakpoint reuse, okay? And we wish to assess this hypothesis in which we have fragile regions and solid regions. That is even evolutionary. It has a biological meaning because some genes are better transmitted together. The point is that fragile regions are very small, and if we have a small number of fragile regions, very short, the synteny blocks follow again the exponential distribution. So we have a second theory that can follow or accept. My apologies. We have a second hypothesis, the fragile model, that can count in exponential distribution, the breakpoint reuse. And the problem is that how can we test the FBM against the RBM? Using multiple genomes. Now, recent studies, we have evidence of birth and death of fragile regions. So, fragile regions move to different locations in different organs. And so we change again the model to the turnover fragile breakage model. And now we have a different model that points out where are the fragile regions And the question is, what causes this fragility?